I'll start with just a quick introduction. Myself, I'm Trent Carrier. I'm the Chief Operating Officer at L7 Informatics. And we have a very esteemed group of folks up here who are going to share some thoughts. Sammy Datwani. Sammy is Senior VP of Development in Synthego. Synthego. Thank you. And has quite an impressive history of tools companies that have been acquired. So several of them by Danaher, I gather. So Sammy also has a MS in Chemical Engineering from Columbia, Ph.D. from Johns Hopkins, and is quite well known and excited to have you here. A lot of history, experience in automation and laboratory automation. So thank you for joining us. Delara Motlagh. Sorry.
It's okay.
I only knew everyone by their first names. Is at Trumo and has spent a long time in cell and gene therapy and focusing on kind of a real passion around the market, and I think will bring some interesting thoughts. She has her Ph.D. in physiology and biophysics from Illinois and executive MBA from Kellogg. And Yan Zhang is with us. Yang is the Chief Executive Officer at Mission Bio. Before Mission Bio, she served in roles at Thermo Fisher, and where she led the teams in China and general management roles for reproductive health and microarray businesses. And a lot of product development commercialization efforts across several different companies. And so Ph.D. in biochemistry from Medical College, Wisconsin. So thank you for joining us. So I'm going to just briefly let each of the panelists speak about kind of their company's view on digitalization and some of the work that they're doing, and then we'll start with some questions and welcome any questions from the audience as we go through. If we spur some thoughts you want to expand on or ask other things that we're not covering, please. We have a question box. Just let us know. So, Sammy, you mind?
Sure.
Just do a quick intro.
So, Syndico, we are integrating engineering with biology, chemistry, automation, and data, and bringing that all together. Our mission is to enable life science research from development all the way from the discovery phase to the clinic. And the way we do that is really, there's the what and the how, and what is we make single guide RNA at high throughput. We create our own in-house custom sequencers. We're able to work in parallel and high throughput, and we couple that to our analytical chemistry stack and visualize that with data. And so that allows us to rapidly iterate and sort of build on that initial platform. It allows us to change what we're making, types, lengths of the oligos. And we've also got a separate platform where we're engineering cell lines. So we're basically utilizing data, whether it's electrospray ionization from mass spec or next-gen sequencing. So we're really coupling it, putting it all together, and helping to sort of rapidly iterate, visualize what we're doing, and that sort of translates to our operations, and doing this at a high throughput, at a high scale, and really enabling the industry by providing these advanced genome engineering products.
Thank you.
Sure. So I'm with Trumo, Blood and Cell Technologies, and Trumo touches the market in a unique way. So we are a solution provider in manufacturing for cell therapy manufacturing. We're also at the front end of the process with apheresis collection. So we really touch the cell and gene therapy care continuum in two places. And so the data component becomes really important in how you bridge not just the product, but all the data and analytics that go with that impact your process.
Thank you. Yeah.
Great. So Mission Bio, we're actually a platforms and solutions company. Our platform is capable of analyzing at the single cell resolution, really combining DNA changes of the genotype with protein-based phenotype changes. So we have actually leveraged the platform's capability and developed dedicated solutions to support comprehensive, deep, high-resolution characterization of your cell products that are engineered. So our delivering to customers really include, you can get the total solution, including the platform and the reagents. We also have what we call pharma assay development services. So our bioinformaticians and our lab operations can actually help to design the bespoke assay specifically for your particular therapeutic products. And our touch points right now mostly are both in your development preclinical, where we can help to accelerate your development effort, through CMC, where you can use our characterization tools for really lot release and accelerate that. And also on the back end, when actually in patients, to continue to monitor, ensure there's safe future for these patients. And not only they're cured, but also they do not have other adverse events, such as potential development of cancer throughout the rest of their lives. And so very excited to be here and very excited to partner with our ecosystem as well as our drug product producers.
Thank you all. So I'll start with the first question, and obviously your companies are all very advanced technology developers. And one question I had to kind of kick this off is, what role do you see technology development playing in the roadmap of innovation for cell and gene therapy manufacturing? How does the work that you're doing move CG&T manufacturing forward in terms of either scale, cost, efficiencies, innovation? Sammy, do you want to?
Yeah, I think it goes back to that comment of building, testing, learning, and really being able to iterate quickly to integrate new technologies, adapt in real time. So I think it plays a really important role.
Mm-hmm.
When I think of our role as technology providers, what the technology is really doing is enabling the science.
Mm-hmm, mm-hmm.
And the science continues to move really quickly. So what we all have to do is make sure that we understand kind of the end in mind, but all the processes in between. So as you're designing, whether it's a device, a diagnostic, different testing panels, data, and all of that, how do you connect all of the different unit operations, the moving pieces, in order to be able to handle a variety of cell types,
mm-hmm.
and to be able to be customized. So there's themes of flexibility to allow the scientists to take those cells and make them powerful, and then to put them into a scalable manufacturing process. So all of the technologies enable that. So I think that's really the role that we play, is having an ear to the market. What are we trying to do? What are we asking these cells to do? And how do we create an environment that they can do that? And then capturing the information along the way.
Great.
Yeah. I totally agree. I think it's really about the science, right? Cell and gene therapy as an industry has evolved so rapidly, and there's so much that is unlike the traditional therapeutics, right? It's not pure, right? It's extremely heterogeneous as we go to produce that. It's irreversible. You either do in vivo or ex vivo. Actually, whatever you transfuse into the patients, actually they continue to divide, they continue to change. Mm-hmm. So it's nothing like any of the traditional chemicals we can think about, and there's complete new definition of potency, purity, and now we have new terms, right? Persistence and many other forms. And so I think the industry really has to come to grasp with what we're working with here, and the innovation all fronts really has to come together. And so we're excited here, but there's so much learning, right? Steep learning from our end about characterization of single cells, but then we need to understand what the industry is looking for. We need to understand how it actually ties in with manufacturing. And so I think this is a great forum for us to educate each other. And that's without that, it's going to be difficult.
Mm-hmm. So, and that's a great segue to my next question and really the focus today, which is data. So how is data, the shape and the type of data as new technologies are developed and brought into process development or manufacturing, how is that changing the demand and the load on analysis, data collection analysis? What does that look like, and how are you guys thinking about supporting customers that are trying to take your technologies forward in their operations? Sammy,
So we're earlier in the ecosystem. So one of the things I think about data, that this is the piece that's missing, is there's standardization around that. You have a lot of bespoke systems that aren't necessarily talking to each other. Way back when in the semiconductor industry, a group of companies sat down and said, we're going to standardize and we're going to do things in this way. So, their chips could be functionally working with the other environment in that ecosystem. I think the piece that's missing here is that standardization and how it all fits together. Otherwise, you've got kind of the wild, wild west where people are doing what they want, right? Ultimately, they're trying to solve the scientific problem, but how do you get there? How do you get there faster? We've got to work together to do that.
I think that's well said. When you think of, I often, in a very simple way, say all these tools need to play nicely in the sandbox together, right? And so that integration of information, and if you look at the ecosystem of cell and gene therapy within manufacturing, as well as where the patients are being treated in the hospitals and clinics, there is that information flow is not there. It happens in pockets, but it's not continuous, and no one's benefiting from that, right? The developers aren't benefiting from it. The patients certainly aren't. And so there is an opportunity within each step of that process to make more connections between the systems, but ultimately that data, there's the data of the cells themselves, but then there's the unit operations and the process, right? Whether it's at the, when you're collecting a cell, what does the profile look like as you're modifying these cells? How do they change? What does that mean? And then ultimately, at the end when you release, we pay attention to these pieces, but when you connect it all, and as we start to advance these processes, being able to marry all of that, right? The patient data, the process data, the performance of your process, that's where you're really now able to start to realize some efficiencies and benefit from labor all the way to having a better product at the end.
Mm-hmm.
So I think it does go back to the biology, right? I hope the field is not scared of the unknowns, right? Because we're dealing with biology, we're dealing with live cells, we're dealing with the processes that we don't necessarily understand. We're dealing with the heterogeneous cellular system where maybe we're a little bit afraid of understanding integration. Maybe we're a little bit afraid of really characterizing your cell products. We're a little bit afraid of just not knowing what the biology means. But I also do think at the end of the day, we also need to be truthful to truly understand what we're working with, because that's really the only path forward. If we're afraid of the truth, or afraid of the biologies, I don't think we're going to collectively generate the most productive and the most safe products putting into our patients. And so is it a big challenge? Absolutely, right? We don't understand all of it yet, but what we're hoping is that with all of the just think about how far we've come, right? AI and big data and analytics and the cellular tools. Let's work together to help the field fully understand this new modality of therapeutics and how they continue to change. So I think there's a lot more support than we've ever imagined, and so hopefully no one is scared and afraid, and I think that would be important.
So extending on that, what do you see are the bottlenecks, within the industry, within partnerships, within data models? What do you see as the bottlenecks to being able to start realizing that connectivity?
I would agree with Sammy. I think a part of it is a standardization, right? Is that can we come together? I think there's a lot of desire that has been talked about in the past couple of years now. That there's a desire to standardize, but I just don't think we're there yet, right? Where everyone is busy with advancing our own therapeutics and doing what we need, but I actually think maybe this particular market condition right now where each company has to watch very carefully about our capital expense, about what we're doing, it might be actually an opportunity for us to accelerate that standardization. Because then with standardization, we don't have to reinvent the wheels within each company. We don't have to go and invest more looking at things that maybe it's already replicated and developed elsewhere. So it's really, I hope that through these interactions, we become bolder and more innovative and embrace the standardization and share more about your experience with the rest of the community. So give us a chance to help you to standardize. Yeah.
I think standardization becomes tricky, right? We say that a lot, and there's a lot of different ways that you can translate and define standardization across the process. One of the challenges to it is that a lot of these processes have intellectual property tied to them. So there isn't a willingness to share.
Yeah.
However, there are some things that are more table stakes that I think can be standardized, can be shared, and that will help elevate the whole industry. And there, I think it's happening in pockets. We can certainly do that better. There's also, again, there's different platforms that people use, and so having them be able to talk to each other and having a broader information highway helps to tie it.
Have a common set of tools.
Exactly. Yeah. Or even if they're not the same, there will continue to be innovation in different tools, but can they at least work together, right? How can you make sure that they work into that workflow so you're not putting that burden onto the end user? Because it does make it harder and harder for them. And
Can I ask a question too?
If I can just on the standardization piece, but because the standards I think are, when you think of even the most basic, fundamental, nowhere near as cool as what the two of you described on assays, but just something as basic as cell viability, right? So fundamental to the process. Everyone does it differently. And so, how do you say that 80% viability means the same thing from company A to B? I mean, there's some really low-hanging fruit that we can work on standardizing that allows, without jeopardizing some of those bottlenecks and roadblocks, if you will, that will help. Please go ahead.
So who is in the best position to take a lead in establishing that?
Which part?
Standardization.
I mean, there's organizations like Allotrope that are doing that. You get a bunch of people in the room to agree on a standard, and you walk out, and you can have that same conversation over and over again for years, right? And I think that's the challenge. Is really it becomes a matter of where do you begin to get enough critical mass to have value, rather than it just being a conversation about standardization. I mean, there are organizations trying to do that. I think, at least our perspective is. The first one to solve the problem is the one that sets the standard, right? Yeah. So nobody was, setting standards on Adobe on PDFs until Adobe set the standard, and now everybody has PDFs the same way. And so I think to some degree, it just needs to, the market needs to move, and whoever breaks through that first and gets enough critical mass, then that begins to kind of drive everyone towards that goal. Otherwise, you're never going to get 50 scientists to agree on the same way to do cell viability. They're just not, right? It's special for their cell type, and it's special for their process and their own experiences, and so that becomes hard to do. But that's it. At least you asked the question, so I can
So I agree, Trent. One comment would be, then we're waiting for the winner, and that's always tricky. And I think that the societies that could set the standards just get the conversation going, and it doesn't have to be, this number or that number. It's really more about where should the line be. But you're right also, because there's so many different companies with so many different technologies, and in angles. If you think of, looking at confluency for iPSCs and imaging that and trying to move away from a human making that call to machine learning with AI, that's not, it seems like it's a solved problem, but it's not, and it's not a standardized solved problem. I mean, we all use Excel. It'd be nice if we had a tool that said, okay, just drop into this, platform and here's your answer.
Yeah.
But I do think you have to wait, maybe not just the winner, but you've got a few leaders that are saying, okay, let's get together, and that's what happened in the semiconductor industry. They were the leaders. They were not the small startups that have this bespoke solution that they're trying to make some money on. It's we're going in this direction. How do we all go in the same direction and row together?
Yeah, and I think, a very tactical example of this that we could tackle now is connectors, right? So how do you get access to your data? If you want to talk about standardizing the data, I would even say you start back with a very basic, how can end users actually access their data dynamically? And if people remember, and there's still a big initiative like tubing connectors, and how do you make connectors so that a bag can connect with a Sartorius bag can connect with a Millipore bag? Like, that's a big deal, right? But it's the same problem with data. Like, how do I connect, your instrument with your instrument, and pull that data out? And actually, data access is a real challenge. So at least that's what we see as that, system trying to pull the data. We're writing custom, code for every single instrument that shows up in a laboratory, because no one, and in many cases, they're not even accessible or documented systems. So how do you guys think about that? Is that a priority for your companies in terms of data access and the tools you build and technologies develop? Have you thought about that? And anyone in the audience, if you thought about that, like, what is those? A lot of organizations are, just their whole business model is just building connectors.
I'll say for us across the whole manufacturing process, we're designing our devices to be smarter, to be able to manage data, to be able to connect in a way that allows our customers, because they're very concerned about that data, right? Cybersecurity and so on. But we're making a concerted effort to have that available. At the same time, we recognize that it needs to connect to other things in that environment. So we are exploring how can we have more open connectivity, so that you put some of that control back into the user's hands on being able to connect the different pieces. Because like you, it's hard to predict what exactly they will be using. So giving them more of an open access allows them to be able to manage and to customize that as they need.
Yeah. Any thoughts?
So we do so really by working closely with, for example, our CDMO partners, right? Because that's where they are aggregating, whether it's supporting trials or actually ultimately supporting providing therapeutics to patients, and certainly have a rigor in their standardization in providing GMP grade final products. And so we definitely look forward to continuing to understand in their world they live in, what analytical tools need to interface with everything else that they do. I think it comes with really the community working together.
Yeah, I would also echo that. I think we need some small group needs to get in a room and think about, okay, this is what we want to get out of it, and we want connectivity to look like this. I see this and you go back to the, whether it's you're talking macrofluidics with connecting bags, the same problem exists in microfluidics. The same problem doesn't exist in electricity, right? We have standardized connectors. So there's an analogy, and the pace of innovation is so fast, and so some of those things are getting left behind.
Yep, sure, yeah, absolutely. So maybe I'll switch. Anyone else want to ask questions or kind of have comments to add on around standards? And yeah, go ahead. Yeah, sure.
That's cool.
Yeah. Talking to the cue.
My question's going to be around something that I think all companies grapple with. The theme I've been hearing here is resources, labor shortage, skill sets. I thought maybe you could discuss some examples that you've seen where companies have allowed their staff either full-time or part-time roles that were around data management for the company. What did that look like? Do they have a single person that's responsible for data science across all departments, or something more like each department has, a subject matter expert for their data? Right? So I thought maybe you could talk about how companies provide resources to do these things. Because I think I heard from you, everybody's so busy just trying to get something out the door. But you also have to have resources internally that are kind of more focused on everything we're talking about.
Yeah, so maybe I'll start. So at L7, we interact a lot with those organizations that are trying to do data intelligence, and so what I see is everybody, at least who we talk to, want to be data smart. I call it data smart, right? But really, it's a matter of there are roles in chief data, or head of data analytics in manufacturing. We've seen that with several manufacturing MS&T teams where they'll have a data person. So we are seeing that kind of, but it's more particular in the cell and gene therapy companies that we're working with, where data is very much being driven through a coherent strategy. The failure we see is when those people sit in IT rather than sitting in the operations.
Yes.
So if those roles sit in IT, then the adoption of the technologies and the receptivity to actually bring in the infrastructure or the change the way of working, you get a lot of pushback, and kind of adoption becomes your challenge. But if those roles sit within the line, to say within MS&T or within operations or within the laboratory, they've been pretty successful, and one person can make a pretty big change in that. They're high demand people, right? I mean, everybody wants to be data smart, and finding those folks is not easy.
For us, it's a hybrid model, and it basically depends on what the data is. And we've also invested, so we have a VP of data now. So we're starting to get that single source of truth. The data is flowing in a pipeline, and we can pull information. But for the chem informatics, it's a different kind of data. It's mass spec, electrospray ionization. It's lots of data, and it's frequent. So we need to get that information to make those decisions. So that kind of spawned out of a chemistry development group that somebody decided, look, we need this, so I'm going to learn how to do this, and figured it out. And that person now owns a very small data science team focused on that, and that we can pull out the analytics and visualize it and understand where we're making money, where we're making more money, where we're not making money, things like that, what the yield is, how fast we're getting product to people that are ordering our products. On the other side, the bioinformatics, it's much richer, and so you kind of need a little more subject matter expertise. I joke because there was a gentleman that I interviewed, and I won't provide a lot of details, but when we talked about a director role, the salary was enormous, and I was just shocked. I was like, wow, because you're competing against the Googles, the Facebooks in the Bay Area, and so if you're a scrappy startup, that's a no-go. So we have a hybrid model where we're trying to centralize it, but until then, we have a lot of information that we're pulling, and we want to keep those trains running.
And it's funny because I think data is the new gold or the new oil or whatever analogy you want to use, and so those people are very highly sought after. It's just how do you utilize them. So for us, we have people that think about data that are building the tools to capture the data, right? Those are the software engineers that are very focused on building. What we've also done is we have a group of folks that are data analysts that really take that data and kind of translate it into the so what. The challenge is that there's a lot of information out there. It's how do you make heads or tails of it, and then how is it applicable? So for example, we have a dedicated group that looks at analytics. We have over 40 years of data performance on cell collection alone. And we can take that and say, what are the different puts and takes in order to optimize and to help standardize such that you get a more consistent collection? And then we can work with individual hospitals and pharma companies to help them customize their SOPs to ensure they get what they need. Similarly, in the manufacturing, we can take performance data, right? So we've done this with our Finia platform. It's at the very end of the process, it's fill and finish. So look at the performance of that system, but also you tie together the customer's individual data, and you can help them develop customized dashboards that shows them, where is there potentially a bottleneck, or where is there a process challenge, and where is there an opportunity. So the data can be powerful depending on how you want to use it. But I don't think it's one group analyzing the data. There has to be a collaboration so that the impact can be seen.
We have invested definitely in our bioinformatics group, right? Because as a multi-omic single-cell tools company, also for us to understand our customers, we definitely invested in them. The interesting one I have to say, so we have some really fresh grads with postdocs. It's amazing. I constantly get amazed by, I guess it's Gen Z now, their sophistication in communication, their capability in understanding science, and their education level. Totally blown away by really the young grads. They've done incredible things for our company, and we did allow remote, and so actually have teams who moved back to India or China or for family reasons, and so we're very flexible. And I think once you create a culture for the company, you are able to hold on to these incredible talents. The other source of talents we had was actually from academia, and some academia core lab, bioinformaticians actually are curious about the industry, and we also it's a great combination. And so, also push your vendors, right? Work with us. You can push us and say, can you guys do more for us, right? So you can combine the different specialties from different partners to achieve what you need to do. You don't have to homegrown every specialty to make it work within your company.
Thank you for the question. I would do a little switch here and talk about how data gets used and analyzed. So, there's been a lot of discussions about AI, machine learning, and applying that in more discovery roles, kind of capacity. But it's interesting to start asking, how does AI, machine learning, how relevant is that for manufacturing, particularly in a regulated environment? How do you create a data learning or a data smart process, but also a regulatory compliant process? Have you guys, have you run into that push and take? How do you manage really applying the most innovative data analysis, but still maintaining a state of compliance?
I think that the technology is still evolving.
Okay.
Right? I don't think it's here and now. The considerations, as you pointed out, right? It's having something that, if you think of sensing, for example, right? Sensors can give you different analytics, but then if you want a feedback loop for the machine to take an act, then you really better know what that means, right? So as we understand the process analytics better, then you have to really program an if-then. And then you have to validate it, right, in order to be able to use it in a GMP environment, and to be able to demonstrate and give people confidence that automatic behavior is going to be what they need it to be. And also, right, I think when we talk about AI and we talk about the being able to fully automate and let it be a smart machine, you've also have to be able to have the ability to program it and to customize it. It can't be fully kind of on its own.
Right.
So these are the pieces that I think the industry is still grappling with, and how do you then make that, how do you enable such a system, but then how much do you really know about the process to be able to make it smart enough to do its job?
Okay.
I'll add the documentation piece is also tricky, because once you flip over to handing off the control to your algorithm, what if something goes wrong? And so it seems like it's more of a push. In operations, it's not a pull to automate this, because the way they're doing it works, right? So ultimately, when you get to that point, you have the information and you can drop that in. And sensing is a good example of something that's, we can jump into that environment and have all these measurements for ourselves, and we know when something's going wrong, because it's, the lactate level is going up. At the end of the day, it's really a question of, you almost need that group in between to massage so it goes through and it stays smooth. And the piece that you didn't mention was documentation. I feel like, documenting those processes and making sure you can flip the switch, and sometimes you run both in parallel and sort of see where their savings are. It's still not there where I think it could be, but also a lot of it is, go back to the example of, if you wanted to just look at confluency of IPSCs, that seems like a solved problem. If you go to the scientific literature, you see what people are doing in academia, but it's not a simple push into manufacturing, because the human eye is a lot better than those algorithms. And so maybe there's a window where you have the hybrid model, and then you can actually flip over after you build the confidence.
In the back.
I'm curious about whether the digital analytics and in-process controls will lead us to batch production records that are operator independent, and whether you feel that it's important to have that biology nuance from the operator in process control. In other words, people are very anxious to collect data during process runs and limit the amount of lot release testing that's required. And I think part of what's important in there is to have assays that begin with high confidence that aren't operator dependent. Cell viability is a very good one. So do you think we're reaching a future where we can just step back and the process will run itself?
I think there's a kind of, there's both a logistics, my experience, there's a logistics and then there's a science problem in both of those. One is a lot of those assays are still, being done, the more complex assays are being done in the QC lab, not in the manufacturing suite. And so we go back to that data highway challenge, which is, even if you do have those assays, how do you actually make that data accessible to the people sitting in the manufacturing suite to be able to bring that data back in, currently even holding numbers up against the sign so they can write them in the batch record is still state of the art in many manufacturing environments. And so the move to that digital highway, as we talked about, I think is part of an answer, part of the challenge to making that more a dynamic exchange. I think the other is that if you look at well-characterized biologics, monoclonal antibodies, right? Your ability to characterize your product and the variations that your process creates in your product, your analytical characterization actually exceeds the range, or is broader than the variation you get out of your process, because that's been run enough where you're analytical, you can characterize anything coming out of that process. If you look at a traditional vaccine, those are kind of on the same order of magnitude, because you're making a process potency assay, you're infecting the cells, and that's your potency assay, that's also your manufacturing process. So those are on the same order of scale. For cell and gene therapy, the process variability is a lot bigger than the analytical capacity to measure it, in part because every time you run an experiment, every time you run a batch, it's an N=1 experiment from what I've seen. You're never in a 14-day process going to have exactly the same process. And so you get a much bigger load on your analytics. You know, the data is the product more than the process is the product. And so as we get more experience, I think it's really about getting those runs and getting the experience enough times to build the data set. I think that's the challenge for the analytical side of it, is you need more data sets in order to start closing the process variability so that your assays can cover that range. Today, you know, you have so much range of potential outcomes from a cell therapy process that you're never going to have the same thing happen 3 or 4 times over and over again, and so that ends up putting a burden on your analytical characterization, and we're just not today, you know, have enough of a data set to learn from that. That's just my experience. I don't know if you guys have it.
Yeah. I think I love the vision, right? I think that really should be something that we all work towards. But having said that, though, you know, I'm not sure about if we're there today, right? You know, there's so much unknown about biology, and I have seen one of the companies, big companies, presentations say, oh, we provide tools for analytical tools for cell and gene therapy, right? They just put the icons of a bunch of different boxes on the same page, and they say we can provide all of the tools. And you look at it, right? There's the viability counter, there's the stain, there's the, I don't know, I mean, flow cytometry, there's all of these boxes, right? I don't know how you can integrate 12 boxes in process QC, right? I think we need to work harder and smarter into consolidating those analytics tools and figuring out what truly we need to measure, and that would have to be step one. And I don't know, maybe I'm being too simple about it, but but I love it. I think that should be, ideally, right? That should be the future.
It is, and maybe while we're waiting for the question, I love the vision too. We're not there, I think, for all the reasons Trent articulated, and I agree with kind of that's a how you can. Maybe a watch out is because of the variability in this process, one of the things we have to be careful about is as we try to fully automate and take out some of the human element, think about the amount of manufacturing failures you may have as a consequence because we're trying to fit it in too tight of a box. So that's the other conundrum that kind of pulls us in another direction as well. These are things I have confidence we can solve as we go forward, but we're going to have to consider all the what ifs when we're trying to put it into these different algorithms.
Yeah, Irene Rondell, BioCurie. So comment on Trent's comment about collecting data from numerous runs. So the other tricky thing is you've got the inherent variability of the process, but also you're trying to optimize the process as you're doing it. So you're essentially, you know, flying the plane while you're building it. So that adds another level of complexity. And then a comment, Sammy, you said, what if something goes wrong? You know, I'm sure if you've heard a lot of talks today, including the automation one yesterday, like for cell therapy, autologous cell therapy, there are over 120 steps. And the probability of a human being getting that right time and time and time again, I think I'd put my money on AI in the machine, frankly. So by the time, once we understand the process and the levers, I think a data-driven process is probably more likely to be faithful than a human being. And you know, I think humans are great for innovation, not so great for doing the same thing routinely over and over again.
Yeah, I 100% agree. I think we just need more shots on goal. I think it's just about repetition. And the basis keeps moving. The repetition's hard. Your starting materials keep moving. It's a complex, it's not a monoclonal antibody you can run on a mass spec and say, here's my glycosylation patterns, right? You got proteomics, you got metabolomics, you've got a whole bunch of data, and then it's not only what is important, but how do I get a standard that I can actually measure against as a reference so that I know if something's right or not, and that process is just, you know, I think quite variable today. We'll get better at it.
I agree.
That wasn't where we were with antibodies 20 years ago. Those were those were art instead of science. Yeah, thank you for the question. And yes, next.
Hi, I guess one of the comment to your point, we need to identify which aspects of each therapy we need to monitor, and one size may not fit all. I mean, the standard metabolite monitoring, lactate, viability, things like that. Yeah, that stuff we need to do for all cell therapies, but there may be some indication-specific or function-specific assays you have to be willing to build in to your process, not only on the starting material, but also at manufacturing. I mean, it's at every major stage of an alteration or manipulation of the cell. To naively assume that, well, it's still the same is the road to hell, okay? Not for us, for the patient, okay? We owe it to the patient to assure the quality of the product. It'll also be a real opportunity, though, to optimize making a better product. There's different ways that data management works. One is to improve productivity or yield, or efficiencies of the process itself. But the larger issue still out there is the quality of the product. What percent of the CAR T manufacturing run are truly API versus what percent of those cells, while they may have the CAR construct, have signal transduction liabilities due to the age of the donor or the manipulations that were carried out.
Yeah.
We're still the sun is just rising on this day. And the reality is we're going to have to build those systems in. We can't avoid it because our immunotherapeutics in our own bodies cure us of cancer every day. But CAR Ts don't cure us every day. Why is that? We should ask that question every time we see a PR instead of a CR. Now, there's a lot of biologic variability. I've worked on cancer all my life. So there's inherent variability in the disease stage, health of and all that. We got that. But even so, people that have the flu cure themselves of cancer. Now, admittedly, the tumor burden is microscopic, and therefore your immune system manages it. We have to back into aspiring to that. I may never get there, but that's not the point. Not getting there is not the point. The point is there's still a lot of room for improvement. And building in insightful assays in real time at major points in the manufacturing process that each person has to figure out. Okay, I'm going to do a 40-fold expansion. You might want to check them. I'm going to do a lentiviral or a CRISPR edit. You might want to check them. Just to make sure you're okay on the downstream pieces that the cell's supposed to maintain, so the CAR construct will work optimally. And I'll get off the soapbox.
Thank you for that. Well, I think it opens a great question, which is, what is really the impact for digital transformation and right now? Like, what's the short-term gains that we see coming? Right? You've talked about quality as being one. Maybe it's scale, maybe it's efficiency. What do you think is being really the short-term win for digital transformation in manufacturing? You guys have a thought?
Wants to start?
It's a hard question. Audience, I mean, what's you're here and you're here? Are you here?
Don't throw people under the bus now.
Look, I think there's a lot of benefits, and if you think of the here and now, there's, I would say, first, looking more holistically at your process, and you made some excellent points. We are guessing a lot, and we're waiting until the end, and we're not even sure. And there's some hesitancy because of, well, what does this mean then for my comparability? What does this mean for the viability of these programs? And so there's a lot of work we need to do up front in understanding the true analytics of our process. And that is, I think, where the data becomes extremely valuable if collected well, and that you're asking the right questions. The challenge is, knowing the questions to ask and having the tools to be able to pull it together. But to me, the power in the here and now is having a much better fundamental understanding of the process that then allows you to be able to design the scaling of it. Because if you don't understand it from the front end, then you're just, augmenting a problem versus really efficiently scaling it. And once you get to that point and you have understood your process, the data, I think, also allows you to be more efficient. So first, it's the quality piece. and truly understanding the science that you're trying to manifest in your process, and then how do you then streamline it? To me, that's really the here and now.
And don't get me wrong, perfection is the enemy of progress.
Mm-hmm.
You know, I get it. But as we go through this iterative process for the next year, 3 years, or whatever, you'll eventually set milestones. You will never
love
be
it.
perfect, but you say, you know
I love
what?
it.
This dropped this much at this point in the manufacturing process, so let's back off to 35 doublings versus 40 or 1,000 doublings. Things like that. You'll never be perfect, but
Right.
you can further refine and optimize the quality of the final product.
I love
Absolutely.
it. Yeah. And I think some of the conversations we had with our customers is the characterization of the process and characterization of the product. You know, that's kind of where Mission Bio is at.
We're intertwined.
Intertwined, exactly, and to really truly fully understand, having that courage to go understand your products and have that full characterization and document the data, so you can in the future go look back and say, is there a correlation, right? You know, not collecting the data along those processes of your products, I think it's going to be a misstep for us.
I think what data provides is the ability to dynamically look into your process. And if we are allowed to adapt to get to the right goal, that's a different way to look at, you know, these sort of, you know, discrete data points that we need to hit versus, okay, we are coming at this, we can change things along the way. That's a paradigm shift because it's not how we operate, it's not how the FDA operates. We have to be able to think a little bit differently to get to the goal. And I think that's what data provides. And we need to get enough information to support that. And then I think we'll be in a better place. So I think it's putting us on the right trajectory.
Yes.
For Syndico now, it's great because we can adapt if we see a process that's going sideways and, you know, we're running, you know, 100 different variants. We can look at that and say, okay, we can make some changes. And we can do that in real time. You know, the human body is dynamic. Why not have a dynamic process, right?
Exactly. I mean, now, the scary part is now.
Where are you from?
The scary
I
part
love
is
it.
you can't, you know, once you lock down your manufacturing process and your CM&C section, oh, wait a minute, we found out that 40 doublings is no good. It's a pain. So I hate to say it, got some legwork to do up front on process development optimization. Typically, you like to build little things in, you send in your amendment, you wait your 30 days, and you're good to go. That's going to be too tedious. We need to grow a set and be ready to do things up front that maybe seem like they delay the project, but really they enable, at the end of the day, a better product, which means shorter clinical trial, lower need for numbers to get power, and better benefit to the patient.
One more question?
Yeah, question in the back, sure.
So you guys have touched on a lot of things, especially the iterative, cyclical nature of innovation, standardization, adaptability to what's coming. As we think about, and we may be quite a ways off on this, as we think about standardization and we think about case optimization, and we think about just grabbing data on everything, there's a reason that we look at models, right? That's the simplicity, the ability to understand things versus the overwhelming nature of looking at data. The other potential downside of a lot of data or standardized, really rigorous data is that it limits innovation. So what are your thoughts here on how to balance getting better and getting more consistent without putting up endpoints, edges for the industry?
Yeah, and I think it becomes, you know, your question for me is a question of infrastructure. Because standardization is great, and you know, and those are the measurements of, you know, of what am I doing. But you know, our focus at L7 is really how do you build the infrastructure so you can actually start collecting the data. Like we're not even having the data in an accessible form. How many companies, how many here have Google Drives with data, Excel folders that are buried, right? And every time you want to go look at that, you're going back to that Google Drive. That's a equivalent of a file cabinet in your office, right? And so I think for me, the very first step is about building the infrastructure so that you can access that data, whether it's off of the instrument, because I don't really know what information is important on that instrument. And if it's on a flash drive that sits on a hard drive that sits in a box in a corner, I'm never going to learn from that. And so I think it's about accessibility of data and about the infrastructure to bring that into a way that you can analyze it. And then that's step one, right? And then you get your turns and you get enough runs and you start seeing things that are relevant and you start creating dynamic models and like all of that. But today, like standards, if you can't get access to the data, like I don't know what a standard conversation is going to get to you, right? You're looking in reverse rather than looking forward through the windshield. So to me, I think that's the biggest hurdle, is actually breaking down the data silos that exist because a vendor is building a box that you can't access, or because a laboratory is on a system that's not talking to another system, or those are the hurdles today to getting a digital transformation. The companies we see doing it are companies that need to scale, and they can't scale by adding more people because talented people are really hard and really expensive, right? To your point earlier, right? And so, it becomes a necessity to be able to build that infrastructure, and that's the first investment I think you have to make. The cloud's great. You don't have to put all these servers in, right? People, 2 years ago, nobody was putting GMP data in the cloud. Now, every customer we have are moving to cloud solutions. So lean on the technology advancements and the infrastructure advancements that are happening everywhere else around us, and I think you then begin answering these questions and not backing yourself into a corner on standards. So, sorry, that was my thought to your question. Thank you for asking it.
Yeah, I can't claim that I'm an expert in that. It is very complex. I mean, some of the things that we do internally in terms of process improvement, we've always asked ourselves the question, does it make scientific sense, right? You have to have a hypothesis, and your hypothesis may be right, may be wrong, but at least those are the starting points with a scientific hypothesis, whether it's contamination or is anything else, right? So that's kind of how at least we thought about it, is just that using science, going back to science as a starting point, and go from there. At least that gives you basic set of data that you want to go collect.
It does, I mean, the science is the root of all of it, and having the infrastructure to be able to capture it is important. I think that what I would add to that is there is a nuance to every process and every product, and if you don't understand the puts and takes of your process, it's hard to standardize it. And that's so it's asking the right questions and doing that work up front in order to be able to leverage the information highway and to be able to put the right analytics in place.
I'd say for me, the holy grail is, having a theory, being able to model that theory, and then getting data that backs it up, and then you make the decision of how much do you really need. Mm-hmm. With biology, trying to digitize biology, it's a lot harder than the chemistry space. Ultimately, we need to have that conversation about standardization just to move it because it helps. And the more we think about it, maybe people will sort of take it on, and it's got to be grassroots unless you wait for the winner in the group to say, this is what we're doing. Everybody has to fall in line.
I think we're at a minute left. So any last questions from the audience, comments, anything from this group you have panelists you want to say as we get ready to close the morning? No, I'll just say thank you to everyone thank you for the early morning to the audience showing up. Great questions, good dynamics. Thank you to the panelists. I appreciate it. It's been a real pleasure up here. And thank you to ARM and helping making this possible. So everyone have a good rest of your day.
Thank you.